Welcome to your last week of outdoor data collection. We will be exploring area and distance effects on species richness of understory plants in the context of island biogeography. Biogeography is a field in ecology that tries to answer why species live where they do. Biogeographers look for patterns in their environment that explain the types of species living in different areas around the world. In most ecological systems, the number of species found increases with the area sample, known as the species-areas relationship. Biogeographers also notice that island isolation also influenced the number of unique species on an island. The further away an island was from a continent or another big island, also referred to as a source population, the fewer species that were present. The observations of species-area relationships and the isolation effect were combined to form the theory of island biogeography, which was developed and published by Robert MacArthur and E.O. Wilson in 1967. They developed a theoretical framework to predict the number of species on an island based on island size and island isolation, or the distance from the source population. The model essentially looks at the balance between the rate of immigration to an island relative to the rate of extinction. It predicts a nonlinear relationship between the number of species on an island and the rate of immigration and extinction. To recap, the original theory states that island area influences extinction rates and distance from the source population influences colonization rates. More information can be found in Chapter 22 of your Ecology textbook. Materials needed. You only need a few basic materials for this study. You will need a length of rope or string that measures approximately 10 meters in length, a meter stick or any stick that you find outside that is 1 meter in length. You'll need access to the online version of Google Earth in order to measure your forest patch area in isolation. And lastly, you'll need your field notebook and pencil. Finding a good site. Think about places around your house that are forested, but may be surrounded by agricultural fields, roads, etc. If you live in a city, local parks with some forested area would be great. You can also use Google Earth to help you locate a good site. There's more information in the course online folder about Google Earth. A few considerations about your site. It should be somewhat of an island, meaning that it is surrounded by non-forested areas. The patch should be large enough so you can have at least a 20-meter buffer from any of the edges of your island. And lastly, be sure to find a sampling spot that is representative of your forest fragment. Avoid places that are dominated by a single species that are unlike the rest of the forest. We want to sample the understory that is representative of the forest fragment within your area.
Importantly for this week, we want to find habitats in which we have an understory, so a lot of plant growth happening down at our feet level, but we also want to find habitats that are surrounded by uninhabitable matrix. So if you see here behind me, I have this beautiful old growth Pacific Northwest forest right over my shoulder. We're going to walk in here in just a second, but if we pan around to the local neighborhood, you're going to see that I'm visiting family right now in the Pacific Northwest, and I'm just down the street a block. There's this man-made wetland, there's houses all around, but there's still this really awesome patch of forest. So I have this patch of very natural habitat in an area that is dominated by a matrix or surrounded by habitat that plants might not naturally try to live in. So we're going to go ahead into this deep dark forest, and I'm going to show you a bit about the protocol we'll use to sample the plant diversity in our different habitat patches around the world. We hiked a little ways. We hiked about 5-10 minutes into the depths of this little forest patch. And so we're here, and you can see it's absolutely gorgeous. We have these beautiful old growth trees. We have cedars. We have a ton of maples. We have obviously moss hanging out all above my head. But what we're going to focus on today is actually what's at my feet. So we're going to use this beautiful old growth forest patch as a habitat patch or a habitat island where plants will grow. And we want to figure out how many different kinds of plants grow on this habitat patch. So this is explained more thoroughly in the protocol that's up on Husky CT, but I'm going to walk through how I would go about setting up sampling for my data collection this week. The first thing you want to do, obviously, is find a beautiful place to sample. As I mentioned, I am in the Pacific Northwest visiting family, so I just walked down the road and found this great patch. I looked on Google Earth and I noticed that this is a good habitat patch that's surrounded by other habitat where these plants that are at my feet right now probably won't grow anymore. And so this makes this a great habitat island. Also, my videographer here is my mom. She's helping me out. So make sure that you're coming with someone and you feel safe, and this should be fun. We want you to have fun with this data collection, even though I think we're gonna uncover some really cool key ecological phenomena. All right, so I found my habitat patch. Once we were in the forest, the other thing that I made some decisions about is I wanted to find a piece of habitat that was representative of the understory. And so if you'll look here, we're in a place where we have a lot of these beautiful ferns, which are awesome and really important aspects of this plant community. But if I'm in a place that's too crowded by the ferns, that might not be representative of the natural habitat in this area. So what I've tried to do is find a place that's open enough that I can get a good sample with my transect of the types of plant diversity that we're going to see when we run this data collection. All right, so I found a good spot. The next thing I want to do is find my anchor point. And so if you'll see here, I brought my little sampling bag out, and I'm going to use this nice old cedar here as my anchor point. And what I mean by this is that we're going to be running 4 transects, so in a cross type pattern. And this is going to be the middle of my 4 transects. Each transect is going to be a sample of the plant diversity in the community and will help us get an overall idea of the types of plants we see here. Okay, so I had help, and I have our transect tape laid mostly out. To get my full 10 meters, I'm gonna have to put a little, probably rock or something there, and then I can scoot the tape out to get that full 10 meters. But this is gonna be my transect 1 of the 4 transects that I'll include for data collection for this study. The second thing I'm gonna need is a 1 meter long stick of some sort. So if you have a yardstick at home, that'll work well. I didn't have that, so I just found a branch that had fallen and I broke it to be about 1 meter long, and this is going to help me with my transect sampling. What we're going to do is we're going to use this yellow 10 meter transect as what we call a gentry transect, or a modified gentry transect. And what's really neat about this, Dr. Gentry was a tropical biologist, and he started these forest transects back in the 1960s, I think, and he was sampling tropical forests all over Central and South America. But he got so good at it that he trained a lot of people to do similar transects. And so what's neat about this is we now have data from around the globe using the same protocol to sample the plant community in diverse habitats all over the world. So we're going to use a modified gentry transect because it's worked well in the past, and it allows us to compare our data to other data sets that have already been collected. And so part one of the gentry transect is laying out this main transect axis. The second part of the gentry transect is I'm going to start at point 0. And you'll notice point 0 is right at my start point. It's right at the base of my tree, so I'm going to start a little ways out just so I can see what's going on here. And I'm going to look at the types of plants I see at point 0. That is 1 meter, 1.5 meters to my right, and 1.5 meters to my left. And so if you'll see what I'm doing here, it's a little bit, you gotta use your imagination a little bit, but I'm gonna hold my transect right in the middle, over my point 0. And what I'm gonna do is look at all of the types of plants I see that occur within about a 4 or 5 centimeter width of this wall. You can think of like an invisible wall that goes all the way down and touches all the plants. And I'm gonna see what types of plants hit that transect. And so if you come a bit closer, carefully, we can see actually down here, we have quite a few plant species to be sampled. Note that you guys, it's not essential that you know the exact scientific names of these plants, but at this point, you should be able to recognize that one plant looks different from another plant. And so we're gonna rely on something called a morphospecies, in which in your field notebook, when you find a new plant, you give it some funny name that helps you remember what it is. So if I'm looking down here, this little guy, I'm pretty sure is a little baby maple. So I'm gonna call that baby maple, and that's gonna be my morphospecies ID for every single time that I find this plant along the transect. All right, so at point 0, and I gotta pull this here, at point 0, I have several of these baby maples, but I also have, if I look a bit closer, I have this little parsley looking plant. So I'll include that as a new morphospecies. So I'm gonna call this probably, and I'll write it down in my field notebook, as parsley plant. I also see this leaf here is a bit different. It's kind of hard to tell, but it's more heart-shaped. So I'm gonna call that heart-shaped plant. And that again is a new morphospecies. And so with my transect here, I'm trying to find all the new morphospecies that are on the left-hand side of this transect, as well as on the right-hand side of this transect. And there's some different things over here too that I'll have to document. The other thing, while I'm really interested in what's happening at my feet, we're gonna try to get a representation of the understory. So more or less, look for plants that are smaller than 2 meters or about 6 feet. This works out really well because I'm 6 feet tall. So anything that's lower than me, I'm gonna include in my survey. So if we look over here, we have this fern, and it's actually a little bit outside of my survey plot because it's a little bit too far to the left. But if it was closer to my survey plot, I would include this guy. He's big, but I would include him because he's below 6 feet tall. So I did this at point 0 on my transect, so meter 0. And then what you're gonna do is find meter 1 and complete the same thing. The one trick to this is, if I go down and I'm looking at the plant diversity I'm seeing here, if I see that maple leaf plant again, because I've already counted it at time 0 or point 0, I'm not gonna include it for point 1. I only want to discover new morphospecies. So I'll do that for point 1. We'll keep going on for point 2, point 3, point 4, all the way out to point 10, just describing new morphospecies. And you'll put all of these in your field notebook, and then you'll enter this into our shared Google sheet that we'll use for our shared class data. All right, so the last thing, once I'm done with this transect, that's the hardest part, is learning how to do the first transect. I'm gonna pull my tape in and set it out, so I'll do my second transect, or transect 2. So in this case, I want to do a cross of my transects where the cedar tree is my focal point. So this was transect 1. My transect 2 is unfortunately going to be going right through this fern patch, and that's fine. And I'll repeat exactly what I showed you guys before. One thing to note, we're going to treat each of these transects separately. So let's say I lay out my transect tape out for 10 meters, 30 feet, and at point 0, maybe I see this parsley leaf plant. Even though I recorded it on transect 1, so it's recorded there as the plant diversity that I'm seeing, I'm going to treat transect 1 separate from transect 2. So I'm going to record parsley plant in transect 2 as a new morphospecies for transect 2. And what we'll do at the end is we're going to average the 4 transects to get a good representation of our diversity in this plant community. And what you'll see next, when I get back to my computer, is I'll show you how to get information about the habitat size, so the area of the habitat patch, as well as its distance to other what we call mainland or mature habitat sources that could be influencing the plant diversity in this region. This is a lot of fun. Come out, be safe. You're going to learn lots of cool things about the plants that you're seeing. Take great notes in your field notebook. But I think you're going to have a lot of fun with this one.